Okay, I'm going to go ahead and get started. As promised on Tuesday, I intend not to talk super long today, just so you guys have time to work through some of the Rosalind stuff before we go. They're in Canvas for today. There's a PowerPoint and 2 data files. You can download those later or now, but you'll need those in order to complete the CoLab notebook. And then there's an optional reading that I'll highlight in the PowerPoint. The main point of this week is to start thinking about how do we translate biological questions, biological problems into computational ones. And today, I'm going to talk a little bit about functional genomics. And TFBS stands for transcription factor binding site, and how we can search for those. These are the protein-DNA interactions. I talked a little bit on Tuesday about part of the problem with finding ORIC is it depends on a protein-DNA interaction, right? And that is not strict sequence complementarity. We don't know the language of protein to DNA interactions very well. And so today we're going to sort of extend that issue to other kinds of protein-DNA interaction to see if we can identify potentially co-regulated genes. So this is kind of for the non-biologists in here, or if you haven't had biology in a while. At some level, this is probably intuitive, but gene expression regulation is complex. For all different kinds of reasons that all boil down to different levels of interaction. So you've got protein-protein interactions, right? All of these blue little oval blobs are different proteins. So different cell, different tissue, you've got different potential protein combinations that those proteins need to interact, and they sort of affect each other's conformation, which may affect which binding sites they adhere to or the efficiency that they bind there. Then you've also got protein RNA interactions. So there are the one that my lab works on a little bit. We're interested in sex chromosome regulation, and we know, especially from Drosophila, that there are 2 key RNAs that sort of hold the dosage compensation machinery together. So there's an RNA protein complex that helps regulate the X chromosome. So those protein RNA interactions also help guide where the transcription factors are going to go bind. And then of course you've got the one that we talked about already, the protein DNA interactions. How do those proteins actually recognize what to bind to? You've got all these levels of interaction on top of, or maybe underneath of, chromatin conformation. So how is the DNA folded? We know that the folding pattern of the DNA also affects how things, or whether things will be transcriptionally active. And that's even on another level than sort of this classic cartoon of how is the DNA wound on the histones, right? I think of the histone compaction as almost like a 2D level, and then there's a whole nother 3D level in terms of how these things are packaged inside the nucleus. So complex machinery regulating which genes are being expressed or suppressed at any given time in any given cell. We have developed a pretty comprehensive functional genomics toolbox to try to interrogate these things at all of those levels. I'd say the most recent one is that 3D level where we can actually try to model how DNA is being packaged inside a nucleus. The sort of primary consortium that tries to catalog this calls themselves ENCODE. So they have a website. So I'm not going to get into lots of detail here because I want to give you guys time to look at other things. But this is the ENCODE website. And these are all functional genomics experiments, essentially. The encyclopedia of I'm having a brain cramp. DNA elements. I can never remember where the C ends. So this is the ENCODE website, at least the matrix of experiments. And you can go in here, especially if you're interested in a model system, and find the experiments that have looked at, a transcription factor of interest or a cell of interest, disease type. And one of the things that I wanted to highlight here is one of the main tools in the toolbox is this thing called ChIP-Seq. And it comes in two flavors. One is a transcription factor ChIP-Seq, and one is a histone ChIP-Seq. I'll tell you what those are on the next slide. But you can see that those two kinds of experiments dwarf all of the other sort of approaches to functional genomics, trying to figure out how genes are being regulated. Okay. So ChIP-Seq stands for chromatin immunoprecipitation and sequencing. This is, like I was pointing out on the ENCODE website, the primary tool now for how do we understand protein DNA interactions. Very high-level overview of how this works. If you're doing a histone ChIP-Seq, you're having an antibody that recognizes say a methylation mark on a histone molecule so that you can pull that down. The key to ChIP-Seq is you have to have an antibody that's going to recognize the thing that you want to pull down. Okay, so if a methylation mark, if you're doing a histone, for example, or if you're trying to find a transcription factor binding site, then you have to have an antibody that recognizes that transcription factor protein. Okay, and so whether you're doing the histone modification or transcription factor, you're going to link that antibody to the thing that you're looking for, fragment the DNA, pull down that DNA, so you're basically getting rid of the rest of the genome except what your protein of interest is attached to. So you've sort of isolated from the ocean of DNA just the interaction between the protein and the DNA that you're interested in. Then we put that on a high throughput sequencer and get all of these little bitty reads. And so in principle, those reads are what your protein was interacting with. Okay? And so that should tell us something about the recognition pattern of this protein DNA language. Take all those reads, map it to the genome. So we'll talk about mapping. If the language is confusing to you, we'll get into this a lot more later. How do we map and all that kind of stuff. But you're basically just aligning all these little fragments, trying to find out where they came from in the genome. There are some normalization steps, won't get into, but at the end of the day, right, if you were scanning along the genome, you would expect that if your experiment worked, you should have these piles of reads that came from the places in the genome where the protein was attached to. Okay, that's the basis of this whole experiment. If you're doing histones, you'll get information about chromatin state, right? Was that DNA wrapped on the histone? Okay. If you're doing a transcription factor, you're just probably going to get regions that are, hopefully in expected promoter regions, like a certain number of base pairs upstream of a transcription start site. Okay. So the matching isn't perfect though. So if you imagine all the genes in the genome that are being activated by a given transcription factor, they may have slightly different sequence. So thinking back to the ORIC stuff that we were doing on Tuesday, and you're gonna do for the rest of today and until Monday, how do you find those sites? You have to account for reverse complements, you have to account for mismatches. In this case, what you're doing between finding the peaks where you've got these pile-ups of reads is converting that into a scoring matrix that says how often at the first position of these binding sites do I see A versus C versus T versus G. And I'll show you a picture in a minute that hopefully makes that a little more clear. But this little picture here is called a sequence motif, or a transcription factor binding motif. And we'll zoom in on that in a minute. But before I move on, I just wanted to highlight, and this is the reading that I said, so this Togon et al. 2025 paper is the one that's in Canvas. This little bit, in terms of how do you go from the sequence part, so I've mapped it and now I've tried to call my peaks, that is not trivial. This step, once you've identified peaks, is sort of straightforward, but you can imagine that when you do sequencing, most of the time what you pull down is a few hundred base pairs long. Our transcription factor binding site is only somewhere 9 to 18 base pairs long. So where in that 300 is my transcription factor binding site? Like, that's not a trivial question. So this paper, it's pretty recent. It's cool because it uses this sort of, I'll call it old school, a couple decades of just doing an alignment of all the sequences and trying to get a consensus and saying, okay, at the position, my best alignment, 80% of the time I have an A, okay? Versus some cool machine learning approaches that take in some other factors about distance, centricity of the read. So there's a support vector machine and a deep learning approach that they use in that paper. And it's sort of interesting because they come up with some recommendations. There's a table at the end that says, okay, if you've got relatively short reads, basically if your data is really clean, your peaks are sharp, this old school position specific scoring is the best way to go. But if you've got really long reads or your data is really messy, then some of these machine learning approaches may give you better approximation of your motif. Just this is background information. We're going to use Jasper, and it's kind of for your own information. Where if I'm interested in doing this for my research, where would I go get the data? So today you're going to have to go to Jasper and get data. TransFac is at least as old as Jasper, maybe older, but it's commercial, so you don't get all the access. You get at some level of you get free access. And most commercial software goes and uses TransFac. ENCODE, Systrome, they're based on the same underlying database, just a little bit different approach in terms of how they extract the binding motifs. And then, especially if you work on a model system, there are organismal specific databases for transcription factor binding that you can go look and pull out the motifs. So Jasper, if you go there, you'll get to a home page that looks like this. Really well-maintained website. They've got a nice little walkthrough to help you find what you're looking for. But what's underneath this, the reason it's important is that it's a curated set of these sequence motifs. So they pull the data from all the ChIP-Seq experiments that they can get their hands on. There are other methods of trying to do, experimental methods of trying to find transcription factor binding sites that are more rare than ChIP-Seq nowadays. But they pull that data in as well, and then give consensus or best curated motifs for as many genes as we have data for. So if you go to the Jasper website to try to find a gene, or you can actually Google, if you know the name of your transcription factor, for example, you can just Google Jasper in all caps and then the name of your gene, and it will take you to this page that shows you the sequence logo for whatever your gene of interest is. And then just walking through, what does this look like? So what are we looking at? So the height of the logo overall tells you something about how well conserved or how confident are you that that's an important site. So if you think about generating a consensus, we've got all of our sequence from our ChIP-Seq experiment lined up. And for example, position 6, 7, 8, and 9, there is no variation. In order to be a binding site for this transcription factor, I don't remember, I think this is P53. Anybody know what P53 is? Like what's, it's famous as like, No, I don't know if it's associated with papilloma actually.
It's the cancer gene?
Yeah, it's the cancer gene. So like this is the most widely implicated single gene across cancers. And so if you wanted to know which genes does P53 regulate, like this is the question we're going to address, you could take this motif and search the genome, and it will tell you every gene where it has a P53 binding site upstream, suggesting that P53 probably regulates that thing. Okay. And so what the motif is telling you is that for P53 to bind to the regulatory region, it has to have this CATG at 6, 7, 8, 9. At position 10, it's basically 50-50 whether it's a C or a T. So it doesn't matter as much. And hopefully you get the idea now. So at position 11, it's much more likely to be a C than a T, can't be an A or a G. Yeah, question?
I feel like the CATG, for example, like really follow that whole 2.0 point.
Mm-hmm.
They're going to be based on a ratio of editing, like you said, you have to like C, half C, half T approximately. But why is it not like all the way to the same height that the G right before it is?
Good question. So when you convert from this, which is the frequency matrix, so in the aligned sequences, how often you see that? A couple things. One, the alignments may not be perfect, so you maybe have motifs that only had that chunk. But more often than not, it's because in terms of how you want to score this thing, the way that it's going to work is you've got your genome or your promoter regions, and you're sliding along that genome with this frequency matrix and saying, just like in what you're doing with the Rosalind problems trying to find ORIC, you've got your pattern and your text. So how do you score the match, right? Depending on how far you've gotten in Rosalind so far, it's either a match or it's not. But if you've gotten to the Hamming distance problems, then it's counting, okay, if it's a match, it's a 1. If it's not a match at that position, it's a 0. Well, there's a lot of nuance between 0 and 1. Okay? And so what this is saying by the height is at position 6, we're going to score that on a 0-1 basis because that's a very important site. At position 10, if it's a C or a T, we want to score it basically 50%. If it's a G or an A, we want to score it as a 0. So that's why the total for that column is only 0.5. Okay, so it's like the average of all the possible scores you could get at that position. Good question, though. And that's sort of the simplest interpretation. There are other ways of normalizing based on the region of the genome. So if you took all the alignments for P53 transcription factor binding sites, you might find that they always happen in GC-rich regions, okay? In which case, you might want to score AT regions of the motif differently than GC regions of the motif, just because by random chance, you're more likely to pull some nucleotides than others, okay? So you can get very sophisticated in how you normalize the frequency matrix or the weight matrix. But they all come down to that issue of how am I going to score a match or not, okay? Okay, so we're about to jump over and do the exercise, but I just wanted to highlight when it says go to Jasper and get a PFM, position frequency matrix, that's the button you're going to click to download it. I don't care which gene. It's kind of be interesting if people do all different genes to see what you get, see which different genes get regulated by different transcription factors. But anyway, when you go to Jasper, there's your button. Okay, so the hypothesis is that genes that share transcription factor binding sites should show similar expression. Another way to put that is, your PI or whoever it is that whose lab you're working in, they want to find genes that act in the same pathway or they respond to the same stimulus, or maybe the disease versus not disease or whatever treatment versus control. You want to find things that respond the same way in terms of their expression. So first pass, turning that biological question into a computational question is, can we scan the genome with these transcription factor binding site motifs? Okay. And just so we're not, we'll talk more about this when we get to BLAST, but we can both reduce the compute time and improve our power statistically if we limit the search space. So the way we're going to limit the search space here is, say, only take 1,000 base pairs upstream of every gene. That way we kind of can be confident that we're in the promoter region and we're not scanning oceans of intergenic space that we know probably, at least at a transcription factor binding site, don't influence how the gene is going to be regulated. Or at least we wouldn't be able to predict that it was that gene that it was regulating, okay? So you can tweak this if you want to in the code. Hopefully you can recognize where to do that in the Colab notebook. And this would only be the first step. If you're really going to answer this question, which is a real question that people ask, like my lab, all the time, the next step would be, do we need to go do an RNA-seq study, or is there RNA-seq data out there to sort of confirm our predictions? Because this is going to say, okay, all of these genes share the same transcription factor binding. Now if we have RNA-seq, which actually measures transcription, do all of these genes actually go up and down at the same time or in the same experiments? So let's go over to Colab. Oh, that's not good.
Jeff, I think for that collab, there might be permissions.
Yep. Hopefully I'm fixing it right now. It's just a button I forgot to click. I don't try it again. I may have to fix the. Did it work? Okay. Yeah? Okay. Okay. So this should be really straightforward and run in a couple minutes. The FASTA format sequence. So I've uploaded to Canvas the chromosome 22 human genome, chromosome 22. Let me get it in the right order. And then I've taken the annotation file. These are GFF files. It stands for genome flat file. I think we had these, at least they were in the data for the Unix part that we did the first module. Except it was for the Arabidopsis genome. So anyway, that flat file, if you were to open it and look at it, it shows where all the genes are in the FASTA file. And the RNAs, all the features that we can identify in the sequence, the GFF says where are they. Okay, and so those two correspond to each other. Then the next thing that you need is to go to Jasper and find a transcription factor that is interesting to you. Then this cell should be pretty easy. Right? If you click play and then go down to the bottom, it will ask you for files. And so I'm going to click my... Oh, okay, yep. Thank you. Got to get out of PowerPoint. Whoa, don't start Anaconda. Okay, so in the Colab notebook, I was just reading from the top. I hit play, the wheel starts spinning, and it should ask you for files. So then I do choose files, and I've got the GFF3. Okay, I uploaded the zipped version of chromosome 22 just because it's kind of big. So you'll have to unzip that. You can probably do that on your local computer. It's not huge. I think it's 400 or 500 megabytes. If you remember how, you could also do that once you upload it to Colab. I'm going to let you figure that out. That's good practice back to your Unix stuff. But if you do it locally, fine.
When I tried to access the file on Collab, it says warning this notebook was not authored by Google. Do you want me to run the cell anyway, or?
Yeah, that just means Google didn't write it, I did. And I don't remember what this, I don't think this is my P53 one, but well, it doesn't really matter. So I clicked all three of those files that I needed for upload. And then I hit open, and now you can see it's running the. Let me zoom in a little bit. It's just telling you the progress of the upload. So the GFF3 not very big, it was pretty fast, but the actual DNA sequence for the chromosome. This is a few hundred megabase chromosome. This is the smallest human chromosome. That's why I picked 22. That was the only criteria. Okay, and then it uploaded the frequency matrix for my transcription factor really fast. Saved them, and then it gives some reports about what it's working with on chromosome 2. This should be the same for everybody. 2,834 promoters. It tells you what it's using for background frequencies because, so this is back to, sorry, what's your name? Manual's question about that motif. Let me go to Jasper. Oops. Right. So when you do the raw PFM, you actually get this data here. And so the code in the Colab notebook is converting that into this, but it's doing what I was talking about earlier, where it just doesn't assume that the background rate of nucleotides is 0.25, 0.25, 0.25, 0.25. It measures it in the genome that you have, or in the promoters that you have. And so it's normalized based on the frequency that you would randomly draw a nucleotide. Okay, so it's telling you how the weights are normalized. Based on the PFM that we uploaded, our motif length is 19, threshold 75. So again, going back to the scoring, now we've got this continuum of matches between our genome and our transcription factor binding site. They go all the way from 0 to 1, okay? And so at some point you have to say, yes, keep this as a potential binding site, or no, I'm not interested in that, okay? It's not always cut and dry what's actually going to bind that transcription factor, okay? But you've got to set some threshold computationally. And so in this case, it's set at 75%. What does that mean in terms of biology? Well, we might get some false positives. So we might keep some sites in, like in this list. Now we've got, looks like for whatever gene that I looked at, 2, 4, 6, 8, 10 genes. Oh, this is only the top 10, I bet. Yeah, top hits. Some of those genes may not actually be bound by this transcription factor, or the promoter may not be bound. If we increase it to that threshold to something like 90%, okay, is that only keeping the top 10% of best matches to our motif, okay? So we're way less likely to get a false positive in there. So it's just how strict do you want to be in terms of what you keep versus what you don't, okay? And there's all kinds of biological reasons and sort of funding reasons why, like, you know, but for me, like, this is my priority, that I'm gonna farm these out to grad students to go do the experiment and see if it binds, okay? So as a grad student, you say, well, what did you set your threshold at and how far down the list am I? Am I gonna waste my time here, okay? Okay, and so in some sense it's that simple, right? But you were given this code, and so in terms of this class, what would you do with this? Well, you probably, for this example, don't need to do anything else, because the assignment is for the Rosalind problems. But last thing I'm going to say today, well, at least lecture-wise, is that the assignment is to take the code that you get for the last Rosalind problem, print it out, comment it by hand. So I know the text is too small for you to see. I keep walking over there to do it on the screen, but then I realized for the recording and the online section, I need to do this on my computer. Like all of these, the text here, okay? This is what I mean by commenting, and this I would say is minimum level commenting that I expect to see, okay? Because this is a real, like when I first wrote this program, this is me taking notes for myself. What does this section of code do? When I was writing this code, I already kind of knew, how for loops work. I didn't need to comment this for loop line, okay? I can't judge that for you, but this is like the last thing on your mind if you're really taking a class is, and I mean really taking it because you want to learn this stuff, is am I going to get a good grade, okay? The first thing is, am I learning how this code works, okay? And so print it out, write all the notes you need to be comfortable that you understand how the code works. Okay. Now, the reason I'm having you do this by hand, print it out, partly I find that it helps it stick, but also because I know ChatGPT is going to tell you how it works, okay? And so even if you are looking at the ChatGPT explanation for how it works, having to copy that by hand into a hard printed version of the code is hopefully going to be useful in terms of learning, okay? So that, I haven't posted it yet, but that'll go up, by lunchtime probably, to a place for you to upload that picture, okay? So then you're just gonna, use your phone, whatever, take a picture of the printed out written comments version of Rosalind number 18. So that's the assignment.